Hi guys! At the time of recording this video, the entire world is fighting a battle against a microorganism that managed to take millions of lives since its emergence in 2020. Yes, you've guessed it right. I'm talking about coronavirus, or COVID-19 as most of you may know it. Do you know that this is not the first time the world has faced the threat of coronaviruses? Late in 2002, a small fraction of population in China and Saudi Arabia were infected with SARS virus, which is a type of coronavirus. Thankfully, it didn't get spread wide and was contained successfully by July 5th, 2003. It was contained in just 1 year. Similarly, cases of MERS virus infection were detected in Egypt, but it also didn't spread far. You may be wondering, if SARS and MERS were types of coronavirus, why only COVID-19 turned into a pandemic? Truly speaking, scientists are still looking for an answer. Maybe it has to do with the rate at which this virus spreads. Maybe it is the ability of people to travel more frequently and to far greater distances compared to the past. Some are even saying the structure of virus might be playing a role in the resilience and contagious nature. One more thing to note here is the fatality rate of SARS and MERS virus was higher, but they did not spread so fast, or they did not replicate fast enough to be spread wide. Whereas the SARS-CoV-2 causing COVID-19 is able to make several copies of it, and the symptoms are taking time to surface, making it easier to spread. The structure of coronaviruses. Here we have the 3D model of the coronavirus. As you can see over here, the virus is covered in a lipid bilayer membrane with proteins embedded on its surface. 3 important proteins found on the surface are the spike proteins, the envelope proteins, and the membrane proteins. Out of these 3, we shall be learning about the spike protein. Spike protein is key for the virus to enter our cells or the host cells. Moreover, it is this spike protein that triggers an immune response in our body, and hence the pharmaceuticals are working around this protein to create medicines and vaccines. The way this protein works is, it attaches itself to the ACE2 receptors that are found in the pulmonary and extrapulmonary cells of the body. So here you can see that we have tried to represent a human cell, that is a host cell that is attacked by the virus, and on the cell membrane there are many proteins found. Even on the human cell membrane there are a lot of different proteins found, one of which is known as the ACE2 receptor. The ACE2 stands for angiotensin converting enzyme receptor 2. And these receptors are mainly found on the pulmonary cells. When I say pulmonary, it means the cells that has to do with the respiratory system, and even in some of the extrapulmonary cells other than the respiratory system, that is like the cells of the heart or kidney or intestine or endothelial cells of the body. Here we'll be seeing what happens when it attacks the cells of the respiratory system. In a healthy person, the exchange of CO2 and O2 gases happens in the alveoli, that is inside the lungs. So here you can see the model of a human respiratory system, where this is the windpipe or trachea, which branches further to give rise to bronchioles and which ends in alveoli or the air sacs present inside the lungs. So in a healthy person, it is in the alveoli, which are clusters of balloon-shaped air sacs where the O2 is transferred from the air to the cells, and the CO2 generated in the body is transferred to the alveoli to be exhaled out of the body. If you look into the inside surface of the respiratory system, let's say we are taking a section of the windpipe and looking at a cellular level, you can see that the respiratory tract is lined by certain cells known as the ciliated epithelium. So on top of these cells, you can see such hair-like structures which are in constant movement. And what does this aid in? It filters the air that we are breathing in. So it is the movement of the cilia that prevents any contaminants from invading further into the respiratory system. Also, there are certain immune cells which are fighting constantly against the pathogens such as viruses and bacteria. One of the immune cells that you are seeing here is a macrophage. So what it does, it eats or it engulfs the contaminants such as bacteria and viruses and keeps the person healthy. So this is the case with a normal human who is having a healthy respiratory system. Now a person who is infected with SARS-CoV-2 virus, we have learned that SARS-CoV-2 infection is occurring more readily in people with prevailing disease conditions, or who are having any prior disease conditions, or who are old, where they have a compromised immune system. So in this case, the virus overrules the body's immunity and reaches the air sacs or the alveoli. There it enters the cells and it multiplies. While it is multiplying, our immune system also gives a fight back, which is called as an immune response, and that causes inflammation, and that leads to filling of fluid in these air sacs. And when the fluid is filled in these air sacs, it interferes or hampers the process of gas exchange, and that results into the person not getting enough oxygen, and that leads to surfacing of symptoms such as difficulty in breathing and fatigue. But the body is still trying to remove this virus by coughing it out. Once inside the host cell, the genetic material of these viruses, which is a single-stranded RNA, begins producing copies of its genetic material using the host machinery, and it also builds more copies of the virus itself. So what we have tried to represent over here is the genetic material of the SARS-CoV-2 virus, which is an RNA molecule. Do remember that there are different types of viruses. There are DNA viruses and RNA viruses. The viruses that have DNA as its genetic material are known as DNA viruses, and the viruses such as SARS-CoV-2, which has single-stranded RNA as its genetic material, are known as RNA viruses. At this point, I would like to deviate to explain about mutations. When building copies of genetic material, a few exhibit slight variations compared to the original genetic structure. This is termed as mutation. Some of the mutations hold no significance in terms of infection severity, whereas other mutation can either increase the potency or how our immune system responds to it. Now you might be hearing constantly the term double mutation. Double mutations are ones where one strain has two mutations together. And like I said earlier, in some cases, this might be doubling the severity of the virus. The purpose of a vaccine is to introduce a weakened microbe or a part of it to trigger an immune response from the body and to create appropriate antibodies to fight against the foreign body. These antibodies are also generated when the actual microbe infects the body. So it could be following any of the below mentioned mechanism. One is where weakened or neutralized or dead microbe is introduced. Now other type of vaccines are nucleic acid vaccines, where an altered DNA or a piece of mRNA is introduced into the body to synthesize a specific protein to trigger immune response. These are considered to be new generation vaccine, as it uses our own protein synthesis machinery to translate a viral protein from an mRNA code, and that triggers an immune response. The vaccines created by companies such as Pfizer and Moderna are mRNA vaccines. There are other type of vaccines known as viral vector vaccines. A completely different virus here is introduced into the body, which carries important information to the host cells. This information is presented on the cell surface as proteins or glycoproteins, and this triggers an immune response. This way, the vaccinated individual is kept safe from the adverse effects of the actual virus. Vaccines such as Sputnik, Covaxin and Covishield are the first type where a deactivated coronavirus is introduced into the body and the body creates antibodies to fight against it. Other COVID vaccines such as Novavax introduces recombinant spike protein. That is, we are saying a part of the virus is introduced into the host body to develop the antibody against it. Pharmaceuticals such as Moderna, in collaboration with National Institute of Health Sciences, have developed mRNA vaccines, where it introduces snippets of coronavirus's RNA, and the host body would use instructions coded in the mRNA molecule to make only the spike protein. And like we saw, when the spike protein attaches to the ACE2 receptors, it triggers an immune response and the body fights back by preparing the antibodies. Now you may be having many other questions about RT-PCR and workings of different tests. We shall be covering this in detail in other videos that we upload. If you like the 3D model of COVID, be sure to check out more of our 3D content and more videos about other science topics. Thank you.